welcome again to our webinar series, and welcome to the first Academic Odyssey Series. These are first-hand talks by researchers on their own journey into academia, and today we are honored to have Ros Gleadow as a guest. So I will make a very brief introduction to Ros, because she's going to talk a lot about herself. So Ros is currently Professor of Plant Science at Monash University in Australia, where she is also the leader of the Plant Ecophysiology group and director of the Agtech LaunchPad. She is a member of the board of Eucalypt Australia, the policy committee of the Royal Society of Victoria. She's also a member of the advisory board for the Agricultural Biotechnology Council of Australia. And of course, she's the president of the Global Plant Council. So thank you, Ros, for agreeing to make the first webinar on the academic series. And the floor is yours.
Good. Well, thank you very much, Isabel. I just want to check sure that everybody can hear me okay and that I'm unmuted and all those things. So is the sound, just let us know if there's the sound as a problem. And I think the light is a bit odd. So I'm just going to try and fix that. That might be better.
Mm-hmm.
Okay, so good. So great. I'm glad the sound is good. So I'm going to start my journey. Isabel asked if I would talk. What we've done in the Global Plant Council is we have these large overarching type webinars. So they're not the normal lab webinars, because we really felt in the Global Plant Council that they were covered by different organizations in particular countries. But we've got things like on international collaborations and science communication and a whole lot of other things. And if you go to our website, you can see more about that. And Isabel thought it would be good to have a series on people's journeys in science. And so having agreed, then she asked me to do it. So here we go. Now, you may have been aware recently there was the International Day of the Girl Child. And quite a lot of people posted on Twitter using the hashtag, "This little girl is me." And I thought, oh, yeah, I'll do that too. So I went and looked through some things, and I wanted to have something about a little child, like not a baby and not a teenager. I wanted something in between. And I found this one of me where I'm about 6. And it's appropriate, I think, because I'm standing in my mother's garden. Also, I'm wearing a frilly dress and looking very, very girly. So who would think that this woman would grow up and be something different? I also note that I'm trying to stand in a very elegant way, which is something I never really got the hang of. So how did I get from this to this? So I'm just trying to hide video panel. There we go. So here I am in the lab, and now I'm a professor. How did this happen? Well, normally people go to school, then they do a bachelor's degree. They maybe do a master's degree, depending on what country they're in. They do a PhD, they do a postdoc. They might have a bit of life somewhere along the line. They do a fellowship, and then they become a teaching and research academic. My journey was completely different. So I did a master's, which was not that common in Australia. Then I worked and I had what I call life. I'll explain that. Then I became an educator. Then I did my PhD much later. Then I worked in science communication and finally became a normal kind of teaching and research academic. So that's my journey. So today, I'm just having trouble seeing all the people. Okay, so today's Odyssey, I'm going to talk about my life and science. So I'm going to do it in parallel, what I've done and how my scientific ideas have developed. I'm known for 4 things, which is too many. I think you would normally say to people, it's better to be just focus on one thing, but I'm not, and this will become clear. The Pathosphere lady, I'm known as, I work on plants that kill, or the Cyanide Lady. I'm also known for working in teaching and educational technology and in science communication. And then I'll end up with some take-home lessons. So the focus of my research is on cyanogenic glucosides. So a lot of plants have compounds that break down to release cyanide, presumably as a defense against herbivores. This is a eucalypt and with a eucalypt leaf beetle, and the beetle also sequesters the cyanogenic glucoside from the plants that it eats. Only a small number of eucalypts have these compounds. Some ferns, clover. I don't know if you remember people eating apricot kernels thinking it was healthy. It's not healthy at all, but they contain these cyanide releasing compounds, pretty much all the plum and cherry family, and apple seeds. So that marzipan flavor you get in apple seeds is actually the breakdown product of the cyanogenic glucosides. And it turns out that about 10% of plants are cyanogenic, and it's actually over 50% of crop plants. So this has been a very interesting focus for my research. And the process of cyanogenesis, I'm just going to run through this. So this is where, in a sense, I end up. These compounds are held in the vacuole of the plant cell. There's an enzyme that's stored apoplastically. And when the leaf is crushed or chewed, they're mixed together. The glucose is broken off. So if we see on the right here, here's the general structure of the sugar, a couple of R groups, and the triple bond of the nitrogen group, the cyanide group here. And then cyanide is released, and depending on what sort of amino acid it was originally made from, you get two different compounds. Now this binds to the cytochrome oxidase, which is the part in your mitochondria that takes up oxygen, which is why people with cyanide poisoning go blue, as you may have discovered if you've ever read Agatha Christie books. She describes it very well. And this is effective in killing animals, all aerobic organisms. Now, there have been some studies to show that it is effective in plant defense. This study wasn't done by me, it was done by my colleagues in Copenhagen. And here is Arabidopsis, which is the lab rat of the plant world, being fed the leaf flea beetle, and it's all eaten. When they transformed Arabidopsis to contain the cyanogenic glucoside found in sorghum, they found it was completely resistant and the insects didn't eat it. So that's good evidence that it is in fact very suitable that it probably functions in herbivore defense. Now, the theories of plant defense are predicated on resource trade-offs, and this has been the kind of theoretical context of which I've worked on. I'll come back to my journey in a minute. So plants, as in any economic system, there's a certain number of resources available, and these are partitioned between photosynthesis and defense, and then that can lead to either growth or the longevity. So it's a trade-off. Do you grow quickly without defending yourself, or do you invest a lot in defense and live a long time, as the classic story? But both of these will relate to how much reproduction there is and how much yield the plants can produce. So what my interest has been how, for example, if you increase these resources, how does increasing photosynthesis affect the trade-off in defense? But then if you limit growth by things such as drought, salinity, and heat, what happens? Do you increase or decrease the defense? So this has been the focus of my work, and I've been looking at plants that make cyanide as a model for studying this. And the reason I used plants that make cyanide is that cyanide has nitrogen in the structure. So therefore, we're expecting it to be more affected by these traits. Now, to go right back and start the story, here I am with my parents. My parents were both medical practitioners, what's called a general practitioner. I'm the fifth of 5 children. And my parents were very outward-going people. I had the great privilege of growing up in a family that valued education. We were all encouraged to do lots of work. The other thing my parents had was a world view. So my father had actually been in the Northern Territory in Australia during the Second World War, and had actually spent a lot of time with the Aboriginal communities up there, coincidentally. And so we had a lot of these stories when I was growing up. And then my mother was also a doctor, which was unusual back in the time. She studied during the Second World War, and she was friends, for example, with Kath Hamlin, who went on and started the Fistula Hospital in Addis Ababa, if you've heard of that hospital. So we had this rather worldview going on. And And I was always very interested in biology. And my eldest sister, when I was 8, she was in her last year of school, and this was her book. And you don't know, if you've grown up now, you don't know how exciting it was to have coloured textbooks. They just had only just come in. And so this book was great. And I think no one had any idea. I used to just lie on the floor of her room, aged 8, and look at all the pictures. And I remember studying all these pictures of these animals. And there was even, one on plants. So of course this book got thrown out, but I found one in a secondhand shop. So I now have my own copy. And then the other thing I used to do that I discovered that was a bit unusual is that I played with dolls. I was a girl, but I used to teach my dolls maths. I used to get my older brothers and sisters maths books and set the problems out of their maths, out of their arithmetic books, and then I would look up the answers and mark them. I discovered that most people didn't do that at a later stage in life. But so it was always natural that I was going to study biology. It was expected that I would go to university. I had that enormous privilege, but I always thought I would be a doctor and that I would work in some underdeveloped country and do good in the world. That was always what I wanted to do. Now, it turns out I didn't do that. And it's complex reasons why I didn't, but probably related to the fact that my father died just the year before I finished school. And I think I just decided I wanted to do what I thought might be an easier road. So instead, I did a science degree. I majored in botany with zoology and genetics. Here am I on my one of my third year field camp. And I decided to go on with botany. In Australia, we have a 4th year of your bachelors, which is called an honours year. It's like a little mini masters. And I did that in botany because the botany people were nice. We did a lot of plant physiology in my study. And I did all the other things that people did. I had relevant vacation jobs. I was in the bushwalking club. I actually spent a time on the student representative council as well. So that was a bit unusual, an interesting experience. And then I got married really young. Andy didn't look like this when I got married. His photos are not that old. Well, I don't know how old it is, but that's when I changed my name. So before that, I actually wasn't Ros Gleadow. I was Ros Hare, but I've only ever published as Ros Gleadow. So that picture there I published as the me at 20 on Twitter. So I don't know if you followed that when it comes around every now and then. So the first thing I'm going to tell you about is becoming the Pathosphere lady. Now, those of you who don't know Pathosphere won't know, but many of you will know this tree. This is a famous Australian ecologist called David Ashton. He was my supervisor, and he had noticed that this tree was invasive in forests and it always clustered in a certain way. So I started as an ecologist, really. And what we found was that under the Pathosphere trees, you had very low biodiversity, but away from the Pathosphere, you had very high biodiversity. Now, I've been watching this now for 40 years, and I can safely say that no eucalypt seedling has ever been observed growing under a Pathosphere undulatum. So if you have a lot of this invading the area, it's going to lead to major changes in the biodiversity. And at the time, the fashion, I guess, in ecology was to study pattern and process. So my project was really looking at the pattern and process of this invasion. And I'll show you a couple of slides to explain what it is. So this is Australia. The green outline is Pathosperm's native distribution, which is defined as pre-European settlement. So whatever you mean, how good or that is or not, that is the definition. And I think at this point, I would like to just pause and acknowledge the traditional owners of Australia. They never ceded their country, and I'm very pleased that I have had the privilege of living here, but I realise that I'm broadcasting this from the land of the Wurundjeri people, and I pay my respects to their elders past, present and emerging. So this orange here is the distribution of Pathosphere relatively recently. And in fact, it's found all over the world, and it's actually very big in the Azores. There's quite a big problem there, Canary Island. In California, it is called Victorian box, if you've seen it there. I've seen it growing on the Stanford campus, among other places. So it's become quite a pest. So what's driving the spread? Is that natural? What's native? Does it matter? Well, the question is, of course, it does matter because it reduces biodiversity. And what it does happen is that this changing in distribution is actually now become very relevant because we know that climate change is driving changes in distribution. So we now actually have to reconsider what we think is a natural plant. So through a whole series of experiments, lots of different studies, washing out roots, which has got to be the worst project you ever did. Everybody always does it. I discovered that the Pathosphere, one reason it was invasive is it was being spread by introduced birds, the European blackbird. It was highly competitive. It was actually more fire sensitive than the eucalypts, but what we call a control burn, or we often burn off the forest here to stop a major bushfire. If that's not hot enough, it doesn't kill a Pathosphere. So it was a change of management practice. So this became sort of standard law. And I said that, you know, we need to have some sort of changed fire regime to control the Pathosphere. And indeed, the Indigenous people likely did this before we came, which probably kept the populations in check. And I got the opportunity to actually test this many years later by actually burning some forest and actually supporting the hypothesis that I put forward in my master's. So that's a long time ago. Now, you think that's all forgotten, but a few years ago, this guy, Jeff Walker, got in touch with me and said, look, I've come across you. I'm very concerned about the Pathosphere in these areas east of Melbourne. You can see there's all this forest, his entire understory, this bit that's been cleared. It's highly invasive. And I had written in my 1982 paper that it was going to be a threat to the national parks and reserves near Melbourne. So he got in touch with me and said, you know what, we should actually do something about this. And so we wrote a couple of little papers on it. And then I got a PhD student. I got some funding from Parks Victoria, this guy, Ben O'Leary. He did a great project. We've written 2 papers in his work. I knew that my ecology theory was like 30 years out of date, so I brought in an ecologist, and so we had a great project. And from his graduation, Ben is now in charge of restoration management for this whole area east of Melbourne. So he's had really great training for doing that. So I guess I still get asked to talk on this every year. Now, then I spent a short spell of time working on wheat. So every Australian plant physiologist does their time on wheat. We are major wheat growers in Australia. And I worked as a research assistant. Now, in some ways, this is stupid, right? I did a master's degree that I wrote 4 papers from. And really, anything sensible, I'll just go back to it. So I wrote this suite of 4 papers. I mean, 4 papers is enough for a PhD, but the rules were that you had to transfer and you had to stay enrolled for a certain amount of time. And I was just kind of sick of it, but it was really stupid. I think I was actually very poorly advised. I should have just written up as a PhD. On the other hand, I escaped from ecology and started working in agricultural plant biochemistry. And it was very new at the time. Gene technology was just coming in. The guy I was working for, Michael Dowling, started the first GMO company in Australia. I worked with plant breeders. I worked with farmers. Very interesting for me. I was a city kid, so to actually work with farmers was a fantastic thing. And it was just a lot of new ideas about how nitrogen was being used in plants, how the protein was transferred from the leaf to the grain. And we wrote some papers on that, and also worked on things that I continue to work on, such as high temperature and drought. The other thing I really learnt through this is that, so I did this for 3 years, was that when you're working with farmers in industry, they need to make a profit. So it's all very well for me to say, it's okay, you know, it's so interesting the way the protein breaks down, you know, from the end terminal and all that stuff, but they don't care. They're just interested in dollars per hectare. And I think that is totally okay. And I really learnt that in this time, the importance of speaking the language of people in industry. So what did I do then? I quit. Now, that just seems ridiculous now. But in those days, that's most Australian women quit their jobs when they got pregnant. I actually didn't have to, but I wanted to. And but after about 6 weeks, I started going completely nuts. And I could not deal with being at home. So I got jobs as a tutor and demonstrator, ended up being the head demonstrator for biology at the University of Melbourne. So this has all been at the University of Melbourne. So ever since I left school, I was still there. I did editing for the Australian Journal of Plant Physiology. I did a little bit of Pathosphere consulting. And this all helped me keep up to date, kept me in the loop. But actually, I was largely at home. Then towards the end of this time, I was asked to develop, I had 3 children in this time. I was asked to develop a new innovative computer-aided learning program for biology students. Now, I deliberately didn't say online learning because there was actually no online yet because the web hadn't been invented. So I love this picture. So this is me with my, 80s perm, except it's about 1990 by now. Here's these funny old computers and our programs that we wrote. And as I do with other things, I wrote a paper on what we developed. And we developed this really innovative program. And part of it was, I was asked to do this by my head of department, Pauline Laudigers. And I just like to give a shout out to her because she's really been this, I guess you call them a sponsor or a champion for me and looked out for positions for me. So I think that that's very, very suitable. Now, I just noticed my light is going very odd. So if you just mind me, I'm just going to give you a little bit of information about what we did, because again, it's relevant to the other things. So what is useful now that I learned from my time in educational technology? I mean, it was quite innovative. I wrote the programs, I wrote the study guides for the students and what they had to do. We worked with educational psychologists, very, very innovative. And I guess what I learned was that good education is good education, right? So you get the structure right and it's okay. And it's, and also that writing code is fun because I wrote all of my own code. But it's a bit like now I've come across the Simon Sinek idea of starting with the why. So often when we teach, we start with what. What do we want the students to learn? But I think we need to start the other way, is why do we want them to learn it? Then we say how we're going to teach them. And then it's kind of later that we think about the what. I think there's a very important thing of thinking about why students need to know a particular thing. So that was a great program. And scientifically, I learned other things too. So we had a section where we were talking about how CO2 was rising in the atmosphere. I just like to add this was 1991 when we started. And that this was going to affect the process of photosynthesis. Here are some diagrams from the program. We looked at how it was going to affect climate change and how that would affect distribution of species. And I came across this great book in the library, this report that had everything I needed to know about it. Of course, I realize now it was the first assessment report from the IPCC. And I actually read it. It wasn't that long. I read it for cover to cover. And in that, part of it was, this is from our booklet, but I made a précis of what we thought Australia's climate would be in 2040. And, basically, it's just exactly the same now. And I think it's just so frustrating that people don't listen, that we knew that then. We knew that we had 30 years to prepare for climate change and really to reduce emissions, and really not enough has been done. Okay, so that was a few years of stuff. And they're still using those programs in different ways. And I'm still using some of the diagrams and movies in my own lectures still today. But I realized that to have a career at university, I would have to have a PhD. So, it was lots of fun having doing a PhD with 3 kids and a husband who was a dean and things like that. But, I took my time. I wasn't able to travel much. I didn't travel at all, actually. And I made the most of meeting international speakers at the national meetings. And I really couldn't have done it except that I had such a good lab group, and for my sisters, and I'd say the sisterhood, managed to do that with my kids. By this stage we're at school. And this is when I became the Cyanide Lady. And so what I was looking at was nitrogen and carbon dioxide and how the interaction of these would change the balance between photosynthesis and defense. And here are the papers that came out of my PhD. And I worked on sugar gums, so Eucalyptus cladocalyx, very highly cyanogenic in the tips, decreasing as the plants get older. It contains the same compound that you get in leaves of prunus and apricots. It's not the one in apricot kernels, that's the diglycoside of this. And you can see here, here's the nitrogen and then the cyanide group. I'm just neat. And so just to cut us, 4 years of work short, when we grew sugar gums at high carbon dioxide, yes, they grew better, they were taller, but you notice that they're less leafy because plants are more efficient when you grow them at high carbon dioxide and therefore don't actually need as many leaves to support the growth. The other thing we found is that if you think, is that these leaves have less protein in them. Because most of the protein in the leaf is from the photosynthetic machinery, that becomes more efficient, and the balance and trade-offs are such that the plants will allocate less resources to protein. So if this is the amount of the nitrogen in the leaf, in a normal leaf at elevated carbon dioxide, it's significantly less, and we see a reallocation of the nitrogen normally in rubisco, in the photosynthetic enzyme, gets reallocated to cyanide. So the leaves have less protein and a relative proportion higher of cyanide when they're grown at elevated carbon dioxide. So this led me to make a whole lot of, get into science communication actually, because everyone was interested, would koalas be able to survive on leaves in the future? And our prediction was no, by 2050 they wouldn't, even though koalas do not eat cyanide-containing plants, just the protein effect. And so we got written up in the Australian, which is now a conservative newspaper in Australia. And we made some videos for the ABC, and I've actually ripped that, put that on YouTube, so you can have a look at that because it's so old now. I still use it for my students and they think it's hysterical. So just to move along then, just a couple more stories. I then did a postdoc on, again, working with industry. So this was working with one of the big essential oil product producers. But I stayed in the same lab. I still had 3 kids at home. By now I had an aging mother with dementia. I had a lot happening in my life. And so I wanted to work part-time and I wanted to work somewhere I knew. And in this case, what we were doing was working with a company to develop elite trees of this eucalyptus to produce better quality and more efficient eucalyptus oil for plantations, because this is actually a natural they normally harvest just natural patches in the state forest, and this was becoming national park and would no longer be available to them. So I had a PhD student, my ex-supervisor Ian Woodrow was my boss then, and we did some interesting work. And so I won't go into the details, but this work's ongoing. In fact, they're still doing it. There's thousands and thousands of these trees that I started selecting, looking at the monoterpene concentration. 1,8 cineol is a pharmaceutical grade eucalyptus oil. And so what did I learn from this? I don't want to focus on this part of the work, but I think I learned, first of all, I do not have the patience for tissue culture. Doing tissue culture just drove me completely mad. There was no way I could spend my life doing that. I learned how to use a gas chromatogram in my sleep. I did so many runs, so many analyses, thousands of them. I learned a lot about working with industry, and again, not dollars per hectare this time, but really they need to be able to make a profit. They need to be able to employ people and look after their owners. But I also decided really, by now I had been at Melbourne Uni since I left school. It was really, really time to move on. So at this stage, I decided to grow up and leave home, which is what I said in my resignation letter. And I accepted a job. I took about 2 months off, and then I accepted a job at Monash. Now, I call it Mission Impossible. This is funny because this was 2005, January 2005, and I got a phone call to say I'd got this job. And the ringtone on my phone was Mission Impossible, you know, when we used to have silly ringtones. And I didn't know how prophetic that was, because this was, when I started, people said, oh, you're doing that job. Oh, you're teaching Sci 2010. Oh, you must be brave. You like a challenge. And I didn't realize that it was, I thought it was a great thing. It was an interdisciplinary unit on how science works. Turns out students didn't want to be there. There are thousands of students every semester. It was resented by academics and students alike with very low student rankings. But I thought it was great because it was what science is. It's actually really affected how I actually do science, because I really understand it more now. The history and philosophy, we did a whole section on science communication and ethics. So I thought it was fantastic, multi-campus. So basically it brought everything I'd ever done all together. And so first of all, I pulled in all those ideas that I'd done with the teaching and got this structured program, questions to focus the work, you know, what the lectures were. I brought in a magician to teach them about pseudoscience, very clear rules about the assignments and how it worked and the dates, color coding the different topics so the students didn't say, what's all this about, you know. So I was really, really pleased. And I did that for a really long time. The other thing I did was really design learning management systems. And my course was described as basket case to showcase in 18 months. So it was really in a bad way. So I've written, of course, some papers on this, but structured the work so students could go to the website, know exactly what to look at. The work was organised thematically, and they could click on the week, find out what was on in the week, and then it would give them all the resources they need. And basically, this is just used everywhere across the campus. It's now regarded, of course, everyone's doing it, but at the time it was very innovative. So so I think that this was a really important thing. So you'd never lose anything. Now, my idea at this time was to start a small research group, because one reason I got the job, because they had actually interviewed some well-known science communicators for the job. They advertised for a lecturer in science communication, and I just thought, oh, I reckon I can do that. So that was fairly bold of me. But they wanted someone who was a practicing scientist. So I thought, well, there's another problem with cyanide, and it's in crop plants. I thought, I don't want to work on eucalypts because I'm in the same city. I want to work on something different. And I had my background in agriculture from my time there. So I thought, well, I'll do that. And there was a problem with heat waves causing cattle deaths. So in Australia, the cattle eat sorghum in the field, actually. They graze on it. It's the leaves are very important. And it contains this cyanogenic glucoside, Durin, and it becomes very toxic. And so I was contacted by this plant breeder, Peter Stewart, to see if I could do some testing. And I said, look, let's see if we can actually develop some low toxic sorghum that might be good for cattle grazing. And so I think at this point, my idea of creating a small group didn't quite come off, because I ended up starting a large group. Here's just some of them over the years, but the particular partners in crime I'd like to mention are Biwa Mola from the University of Copenhagen, who's been a collaborator for 15 years, Peter Stewart, who's the agriculturalist I work with, and Cecilia Blomstedt. You can see Cecilia in all the pictures because she's the senior scientist in my lab. And actually, when I'm off teaching and being president of the Global Plant Council, she's the one that makes it work. And I've collaborated with all these different people. So what was going to be a small research on the side just became fascinating. And I think there's two things that I think I'd like to say about this, is that when I did my PhD, I did it because I thought I can't work in a university without a PhD. And I did it in botany because I thought it would be easier. But what happened was I just loved the research. It just was such a surprise. And I loved the way it sharpened my thinking. And I loved the way you had to be so logical and methodical and the troubleshooting. And it was the same with this research. It really took off, and I just really loved it. So the question was, of course, should the cattle eat this? And droughted sorghum is much more toxic. So there's the cyanide, and if it's droughted, it's much more toxic. This is published in this paper. This paper has CO2 in it as well. So this is consistent with the theories that I've been looking at, where resources, if we limit growth by droughting the plants, then the plants will make more toxins. So it all seems very, very straightforward. So that agrees with the theory. However, if you really super drought the plants, it turns out that that's not actually the case, and they don't actually get so toxic. I think I'll have about another 5 minutes to talk. So we created an acyanogenic safe sorghum using by creating an EMS tilling population. This is the synthesis of cyanogenic glucosides, and this was all worked out by the Danish group. And we used a mutagenesis approach, so the GMO is not acceptable in Australia, and we found a number of mutants that we could knock out the first gene and stop Durran being produced. And we found a number of structural and regulatory mutants, and these have been the subject of ongoing research because it's a fantastic model to study for theories. The other plant that I've worked a lot on is cassava. That's also very toxic. It's eaten by 1 billion people every day. It's cyanogenic, so it can be lethal unless it's processed. These children have konzo. This is a photo from the 1980s outbreak of konzo, where Julie Cliff, a Melbourne doctor working in Mozambique, made the connection between cyanide and this paralysis. So there are ongoing outbreaks of konzo in droughted times. And so what we found, we didn't find, but what had been found by others, is that the World Health Organization recommendation for cyanide in food is less than 10 parts per million. In a normal year in Mozambique, the flour would be about 20, and a drought year over 200. So you can see that this is a major issue. So what we have brought to this discussion is that we know the same thing that we'd found with sorghum, is that if you drought the cassava, it becomes very toxic. So in this case, these are tubers grown in the greenhouse under well-watered and droughted conditions. And you can see here's the WHO recommended safe limit for food. So you can see how toxic it can become. And so this has now got really raised a lot of issues where we've looked at lots of different issues around cassava. So I guess in some senses, I've come full circle. I am actually now a doctor. I'm a doctor of plant science, and I'm working in public health in developing countries. So I think there's perhaps just many, many ways to get to your destination. This photo's in northern Kenya. So we've also been working in Fiji. I won't talk about that, and I won't talk about this, but this is our new theories, because I think we're running out of time, aren't we, Isabel? So, so just quickly, what happens is that to test this, we did a whole lot of serial planting with my PhD student in Fiji, where we looked at cassava planted at different times, and we found that we compared a sweet variety and a bitter variety. And what happened is that so why don't people just plant safe cassava? Some cassavas are safer, but the people very often choose bitter or the high toxic varieties. And so what we think we're finding is that bitter varieties of cassava and many other plants may in fact be more stress tolerant, and that could be why people are selecting them. So it may be more education about processing is needed rather than less cyanogenic varieties. That's open for discussion. So we've been looking at how all these things are looked at, sorry, this is jumping around a bit. We're looking at how the plants take up nitrogen, how that's partition between photosynthesis and cyanide, and the cyanide can be detoxified, used for growth. Plants can remobilize that nitrogen, and this actually uses up energy. Now, I'm excited by this because I can work on Ros, because I'm Ros, but so we've now got these ideas. I won't go into the details, but that cyanogenic glucosides may have in fact evolved for plants to manage their nitrogen and manage their stress just as much as defense against herbivores. And to test that, we're doing a big study of the native sorghums to Australia. Most sorghum, wild relatives of sorghum, are found in Australia, and they're very low in cyanide. And we're thinking if we can maybe be able to use them as a source of material for making safe sorghum. So my take home, this is my second last slide. What would I say to my younger self? Well, first of all, you have time. So I'm actually the first generation of women in Australia who have actually mostly worked through to retirement age. Nearly all women in Australia retired much younger. So that's the first thing. There's much more time than there used to be. Have a go, do what you love, but remember you won't love everything you do. Don't underestimate yourself. Don't be put off by others. Professor Peter Doherty, Nobel Prize winner, says you need to focus and have one thing you're a specialist in. I've kind of done that with cyanide, but I have way too many things. Be strategic. You know, yes, you've got to work hard, but get the runs on the board. It's okay to start small, publishing, applying for funding, get awards. You need to get those things, but you can start with little things. And the other thing I'd like to say is get out and get known, attend conferences, join societies, join networks, and travel. Now, for that, I know a lot of people are wondering if we can't travel much at the moment because of the pandemic, but you know, about carbon miles. Well, when you live on a far-flung island like I do, it's a very big island, but it's a long way away from a lot of other things. It's really important to travel. And I couldn't do that when my children were young, and it's really important for networking. So the other thing is to what's called get a gorilla, get someone who has your back, and do that to someone else. Be a gorilla to someone else. Be generous. Be nice, because you know you keep coming across people all your life, so don't alienate people. And Professor Barry Marshall said when he won the Nobel Prize that it was important to be nice to Swedish people. Finally, the journey, we think there's a goal and there's all these milestones, but in fact, there's many journeys, there's many pathways, there's different goals, there's different ways of getting there. And my kids gave me this picture for Christmas one year, quite poignant. Life is an adventure, not a worry. And it's good to be reminded of that. So, and finally, don't forget to have fun and hear all my grandchildren, and don't forget to smell the roses. And there's the structure. So thank you for your attention. I'm sorry I went so long.
Don't worry, Ros. It was fascinating. So I have many questions, but I want to leave the room to the people attending. So you can write your questions either in the chat box, or you can use the feature that is called questions and answers. While we wait for some of the questions to come in, I will start with my questions. So I have many, so I have to focus my attention right now. So can you please go more into detail how you were would you recommend to develop international networks when you are not able to travel? That is specifically important now due to the pandemic, but it's also important for early career researchers and PhD students for countries where moving around and attending international conferences is not the common thing. So I can speak for myself. I didn't went to any international conference during my PhD, in fact.
So can I just tell you that the first international conference I went to that was not in Australia was only 13 years ago.
Oh, 13 years ago, okay.
13, 1-3, like really not very long. I only went into, but I did make the most of going to international conferences in Australia. So, and this is why I was thinking, like next year we've got the International Plant Molecular Biology meeting, and some people have thought, well, will I travel all the way to Australia? Or hopefully that can happen. But what I'd like to say is if you're asked to be a speaker at this, what you're doing is you're giving an opportunity for these people in Australia who can't travel to all these students. I think that's important. So I made the most of the conferences that were in Australia. And also even then, I often couldn't get away for long. So what I would do with our national meeting, I'd go up on, like, say, one day, and I'd go to all the sessions. I'd go to the society dinner, not the big conference dinner, the society dinner that night. And then I'd stay the next day and I'd go back. So I was actually only away one night. And basically everybody thought I went to every conference all the time, every year, but I didn't.
Mm-hmm.
And so I was very strategic. I'd look at who the speakers were and make sure I would go, sometimes only just for one day, on the day when the speakers. That's how I met Biwa Moller. It's how I met Steve Long, all these Mike Stitt, all these people I met by doing that in Australia.
That's very clever. And I do in Monash, do you teach strategy for the PhD students? So academic strategy? Because
they do now. I mean, of course, we didn't have such things when I did it, but yeah.
So Catherine, we have a question from Catherine. Not really a question, but you really see how the skills you gain along your route are valuable for your scientific career. It's an important lesson in doing what you enjoy and are good at, and realizing that this might vary over time. How much do you think the route you have taken makes you the scientist you are now?
Yeah, I think that's a really good question. And I think, you know, people have their 5-year plans and their 10-year plans. Well, you know, I think I have my 5-minute plan. But I think that's absolutely true. Everything, even teaching history and philosophy of science. I mean, even understanding these sort of how pseudoscience works. And I just found that all of that feeds in. But I think what it is that you have to be open to the experience. So you have to be open to learn from all those experiences and not resent them. So I think, yes, they have all shaped me. And if I'd done my PhD instead of a master's at the beginning, you know, I might have had a PhD in ecology. Well, that would be fine. I'm sure that's okay. But I think what I'm doing now is much more interesting.
Okay. Another question by Catherine. Another lesson from your talk seems to be write up everything. That's something I also noticed. Would you say this has been vital?
Yeah, absolutely vital. So I think you need to write up everything. So I do a little project on wheat, we'd write it up. I do a little project on education. I say, well, let's write it up. Why don't we get it out there? And there's Professor Peter Doherty, who's a well-known Australian Nobel Prize winner. And he wrote this book, A Beginner's Guide to Winning the Nobel Prize. And he says in it, the experiment's not finished until you write the paper.
Mm-hmm.
And I think that's absolutely true. It's when you write the paper and you put it in the public domain, then it's there for everybody to discuss. And even if it's in, you know, Plant Protection Quarterly or the, you know, whatever, it's still there in the public domain and people can access it.
Mm-hmm. Yeah, I was told the same. No experiment is finished until it's published.
Yeah, I still feel guilty because I've got so many. I'm still writing.
So I was amazed when you said, because that's not an option here, that you were during your postdoc, your position was part-time. So I don't know if that's an option for many people in many countries. So how hard it can be when you don't follow the typical path if you don't have these options available to you?
Yeah, so I think there's a couple of things there. One was the grant that we got, which was an Australian Research Council grant, was probably not as big as they would have wanted. So I think my advisor, my boss, was quite happy to pay me, you know, 0.6, 0.7 instead of full-time.
Mm-hmm.
The other thing is that I felt that a postdoc, you know, unrealistically, we expect postdocs to work very hard. Like a lot of, in the US, you know, those postdocs are working 60 hours a week and more. And I knew with 3 children and a mother with dementia, there was absolutely no way that I was going to be able to work more than, you know, 30 hours a week. Mm-hmm. So I did that. I worked about 30 hours a week. Now, if you're in a country where that's okay, then you can be full-time. It depends on what happens is that you end up spending all your time at work on all the boring bits, like all the safety protocols and all the boring things, and you don't have enough time for the actual research. You know, there's kind of like a basic amount of stuff that just has to be done in the lab. Mm-hmm. That is not productive. So I didn't really feel I was very productive. And the other thing I felt like I didn't do is I didn't get enough of that that postdoc troubleshooting experience, because I didn't have time. So I think it has been hard for me. I don't think it's ideal. I think it's better if you can do it full time. And I think the systems are better now, and I think I could do it full time now. I think me at that age could do it full time now, but I didn't understand quite how to do it then. Get a cleaner, I would say, is number one, if you can afford
but
it.
also during the postdoc, you are still learning, so that's almost impossible to know in advance. Yeah,
that's right.
So many questions coming in from Amir Sohail. Sorry if I butchered your name. As a PhD student on the way to being a scientist, we face a lot of obstacles to achieving our goals, but sometimes we start losing motivation. So how did you manage yourself to keep yourself motivated?
Okay, so there's a couple of answers to that. As a PhD supervisor, I call it the second year doldrums. The middle of your PhD, it's very hard to keep students supervised. And what I propose for my students is I try and get them to do something interesting. I had one student that, go to Colombia for a few months, or they go to an international conference, or get them to do field work in an exotic location. So I think that's something you can do, is managing it. I didn't have that opportunity. On the other hand, I knew what it was like to do all kinds of other jobs. And here I was doing a PhD, and I was just doing a PhD, and I wasn't doing anything else. And to me, it was just like, it was just so much fun. Like, I just loved going every day. So personally, I didn't ever lose that motivation. It was just like, yes, this is just the best thing I've ever done.
Yeah, but because you were also older. So it was
I was older. Yeah.
And I didn't make it. I would say you make a more conscious decision than most PhD students, because it's just what you have to do after the master. So you don't think about it that much. But in your case, that was pure motivation.
Yeah, pure motivation. I mean, initially I thought it's a ticket. I have to do it. And actually I wrote up my PhD. I'd done a big field trip, actually in my second year. My supervisor probably knew that. And I hadn't written that up. And I said to him, is my PhD, have I got it done enough? And he goes, you're going to have to write up the field trip. So really I was like trying to do not the maximum amount that I had to do. But in the end, I mean, I published 3-4 really nice papers from it. So it was long enough.
So next question from Kaitlyn Moore. Thank you for a great story and insight into your academic journey, Ros. What advice do you have for applying for grants, especially for early career researchers folks?
Okay, so there's a couple of things. One is that you have to be seen as trustworthy with money. And that's really hard to get. So what I did when I was doing a postdoc, I got little grants actually for doing teaching programs at uni. They had these little internal programs where you could just apply for $5,000 and employ an assistant to put things together. And so I just started with those little tiny things. And on the basis of that, and then we could write some little papers on it, then you show that you can do it. So I started very, very small. So that's one, start small. You need to get awards. I looked around and I thought, I've got to get some awards. I couldn't think of anything I could apply for, because I was like, they were all for people under 35, or, I never seemed to fit the category. And the first award I ever got was for occupational health and safety, because I thought I can get that. So I just, once you start getting them, it's easy to get more. So that's second, you need to get awards. The third thing, and Caitlin, you probably know this, is to collaborate with people. I mean, collaborate with people who you want to work with, but also, I mean, there is that sense of borrowing their CV. I mean, I couldn't have done the things I've done without the collaborations that I have. I could never have done them without the collaborators I've got.
Yeah, so again, think about the strategy, I would say.
Yeah, I think you need to be strategic. So you think, okay, I want to work on this person works on that. I'll go to the conference where that person is speaking. I mean, looking back, it sounds very contrived, but that's in reality, I think that's what I did. I need to meet this person. They're working in the field. Oh, I'm traveling in Europe. I will go and visit them. I'll go and offer to give a seminar at their lab, and then you build up the collaboration. So it takes time to build collaborations.
Yeah, it really takes time.
It takes years, but it's really worth it. And it's so good now because you have friends all over the world.
So we have a new question from Catherine. This one is of special importance for introverts. So for knowing you, I would also say that a critical factor in your career has been your ability and enthusiasm for talking to other people. Again, I think this is vital. You have to learn from others, engage, and show your passion. Any tips for people who find this hard, that is introverts?
Yeah, I think, I mean, collaborating with extroverts is useful. So I think, for example, talking with industry people, I find it easy to talk to people, partly, I mean, big family and all that. So I think that's important. I think so I think when we have there was one project we started working in Mozambique, and we had, I was working with another person who was very strategic in what we did and so on, but I could do the talking, right? Because I was easy to do the talking, and I'd been in Africa lots of times before. So actually through my husband's work, I'd been there. So it was that doing it as a team. I think that was one thing. Not everybody in the team needs or even should be extroverted. And I think if you also think people is to actually care less about how you come across. So yeah, don't care what people think about you. I think that's really important. And I think that's just an ongoing lesson. And the older you get, the easier it is to not care. But I think when you're young, you're very aware of how you're coming across, and I wouldn't worry too much about that. If you think about what does this person need to know? So if you're working with industry, what do they need to know? If you talk, that's the key. And introverts are enthusiastic about their science. They just don't show it.
So it's this connects again with something that you have said during your talk, to understand the language of the people you are talking to. It don't matter if there are farmers, of industry people, of academia. So we have a last question from Amin Sohail, and I think it would be the last because we are over the time already. So as a student, how do we find an international collaborator? I mean, how can we start our journey to make collaborations when you are still a student?
Yeah, I think that's, so one good way is, if it's possible, go and spend time in another lab. So I know in Australia, we have this great program in the Australian Society of Plant Scientists called the Robertson Award. And it's specifically for a student to go and study in another lab to learn a new technique. And that's fantastic. So you need to keep open to those things. That's the bit. The other thing is to, when you go to a conference, you can either ask questions, introduce yourself. Most people are so pleased that there is someone out there actually reading their papers, that they're very happy to talk. And it's sort of, yeah, I think it just takes time.
So that will be the advice. Take the time to make the collaboration.
Yeah. So it might be you meet them at one conference and then the next year. So strategically, for example, I didn't know many people in America and in the US. I knew a lot of people in Europe who had been a fairly regular at SEB. I thought, well, I need to go to the American plant biology meeting. It's a bit more molecular. And so I didn't just go once. I went every year, 3 years in a row.
Wow.
And by going 3 years in a row, then I went 2 years, missed a year, and then went 2 years. And so you keep coming across the same people.
Mm-hmm.
Again, I know they didn't know me. They weren't going to give me a talk. This is like, it's not that long ago, 2010 or something. So I gave a talk in the education section.
Mm-hmm.
Turned out the guy running the education section was a sorghum researcher. I mean, you just never know where your link is going to be. But so I went to the same conference 2 years in a row to make sure that I could build on the collaboration I had. I did the same with the SEB in Europe, the Society of Experimental Biology in Europe. So I would also, one thing I'd just like to reiterate is I think joining professional societies is good. If they have a postgraduate representative, you can be that postgraduate representative, and then they will get to know you and you will get to know them. And that's another way of doing that.
Yeah, I would add here that something that not many early career researchers, I know, I didn't know during my PhD, that one of the most important times to network is during the conferences, but not during the actual talks, but during the coffee breaks and the beer in the afternoon.
Yeah, absolutely.
And probably the lunch, if you get to sit next to someone of interest.
Yeah, I actually used to look up the people before I went and see when they were going to be there and look at their picture and made sure I sat next to them.
So again, the strategic thinking. So let me check the chat box. So I think we have covered all the questions already. Yeah. There are some nice comments still there from Lillian. There is, this is very nice. I hope in future series you will put some soft skills for young researchers here. Indeed, we have done already. We have a couple of talks on science communication, and we are preparing for the upcoming year some others. We are going to have one on ethics. So that's something you have mentioned in your talk. I mean, it's not so obvious to talk about ethics in research when working with plants, but there are some things to take into account there as well.
Yeah.
Yeah, that's so. For thank you, Ros. Thank you so much for your talk and for the time.
Sorry, I really sort of rushed. I think I thought I had the timing right, but then I got too comfortable. So I'm sorry, I took a little bit too long. But
don't worry. And also the questions were quite interesting. So we can think about leaving them there. Yeah, normally, we cut them off. Yeah. So we only post the actual talk and not the Q&A. But I think this is worth posting as well.
Good. Well, thank you, Isabel.
So thank you all for attending our webinar today. Our webinar series will continue